Okay, this is part 6 of the muscular system. We have quite a bit to cover, so I'm just going to get started. This slide is a repetitive one. Remember, we covered this, so now we're going to be talking about the action potential. So, an action potential, we're going to have two different types. We're going to have an end plate potential. Right? Here is our neuromuscular junction, right? And here's our end plate. There's the rest of our sarcolemma and our T-tubules. An end plate potential is what happens here. This is to kickstart an action potential. What happens here is just to propagate it. So what happens here is slightly different than what happens here, okay? So we're going to be talking about both of these, but in general, an action potential is really the same thing. It's just how it's triggered here versus how it's triggered here, okay? So in order to cover those, we have to talk about channel proteins, which we've already done, and this resting membrane potential. So this resting membrane potential just states that there is a difference across the membrane of a resting neuron or a muscle. Okay, so if here's the membrane, it would either be the sarcolemma, right, the plasma membrane of a muscle cell, or an axolemma, the plasma membrane of an axon. There is going to be a difference because there's different concentrations of ions. So the outside is going to be positive, and the inside is going to be negative. So that is referred to as a resting membrane potential. That's the way it is at rest. We have a higher concentration of sodium outside, higher concentration of potassium inside. This is how it looks at rest, okay? So outside is positive, inside is negative, greater concentration of sodium outside, greater concentration of potassium inside. This is what it looks like at rest. And we can kind of see that here, all right? Greater, you don't have to memorize these numbers. Greater concentration of sodium outside, lesser inside. Greater concentration of potassium inside, lesser outside. Right? Outside is positively charged, inside is negatively charged. So now we have to talk about these channel proteins. And like I said, we've already kind of talked about them. So we've talked about chemical gated channels. They open with binding of a specific neurotransmitter. Voltage gated channels open and close in response in a change in the membrane potential. So when that resting membrane potential changes, voltage gates open. And then we're going to have some passive or leakage channels that are always open. So we can take a look at examples of this and this. And let me just say, you don't have to worry so much about these examples. This is just to get the concepts down. We're going to cover these examples as it relates to the process later on. So example, we have a potassium sodium gated channel closed when neurotransmitter is not bound. So these ions can't pass. So when the neurotransmitter, and in this case, we're talking about acetylcholine, since we're talking about skeletal muscle, it's acetylcholine. So once acetylcholine binds to this gated channel, it opens, sodium will enter, and potassium will exit. Right? So once acetylcholine binds, this receptor opens, and we have sodium and potassium passing through at the same time. That's the biggest difference. And so you might even want to kind of note here ions pass at, wow, that was bad, at the same time. Voltage gates, we have one for each ion. Okay, so voltage gated channel example is a sodium channel. Okay, it's just for sodium. Closed when the inside of the cell is negative, open when the inside of the cell is positive. All right, so here is what it looks like at rest. So this voltage gate is closed. Once this patch changes, it's now negative and positive. This gate's going to open, sodium enters. Hopefully you can start to picture how this happens, what is going to transpire here. So when the gated channels are open, ions move quickly. They follow their electrochemical gradients. We're going to talk about that. And really don't worry about that too much. So electrochemical gradients, ions flow along their chemical gradient, higher to lower, and they follow their electrical gradient, positive to negative or negative to positive. So the electrochemical gradient is just nothing more than taking those two together. So this is just the basics behind action potentials. So I've already done this. Here's our neuromuscular junction. We have chemical gates here. So our end plate potential, the whole point of this is that we're going to have a voltage gate right here, and we just need to flip the potential right here. Then this voltage gate opens. That's all these chemical gates are for, is to just flip that resting membrane potential, so then voltage gates respond by opening. Hopefully that made sense. We are going to go into greater detail with that. Right, and that is the end of this part.